Hi everyone, in this video I am going to present a topic on basics of microscope. Microscope is an instrument that is used to see the objects which are very small for our naked eye. It enables us to observe and study the structures of cells, tissue and microorganisms. There are two main types of microscope, compound and dissecting. The compound microscope is having two or more lenses. But dissecting microscopes has a lower magnification and is used for observing larger specimens. Then parts of microscope include eyepiece, objective lens, stage, arm, focus knobs and light source. Eyepiece or ocular lens will magnify the image, and the objective lenses provide different levels of magnification. Okay, then stage is used to hold the specimen. To use microscope, we need to place the specimen on the stage and secure it with clips. Okay, then select the lowest powered objective lens and focus by turning the close knob until the image is visible to our eyepiece. Okay, next we need to focus the fine focus knob and it will make the image clearer. Finally, switch to higher magnification objective lenses and to focus. The next concept is types of microscope. It will include light microscopy. Light microscope, in a sense, it will uses a visible light for examining the specimens. And compound microscope is there. It will uses two or more lenses to magnify. Then stereo microscope, it has two eyepiece and binocular view. It is used for studying larger specimens in 3D. Then polarizing microscope, this is for examining crystals, mineral and other materials. Okay, then electron microscopy is there. It will uses a beam of electrons instead of visible light to view the specimens. Okay, it is of two types, TEM and SEM. TEM in the sense transmission electron microscope and SEM means scanning electron microscope. The next type is scanning probe microscopy. It will uses a physical probe to examine the surface of a specimen. This type of microscope has extremely high resolution. Then types of scanning probe microscope includes. First type is atomic force microscope. This uses a very small probe to scan the surface of a specimen. It will produces a 3D image. Then scanning tunneling microscope. This also uses a electron clouds around the atoms and molecules to create an image. Then acoustic microscopy. This one will uses a sound waves to examine the specimen. Okay, then most important type of microscope is transmission electron microscope. It will produces a high resolution image by transmitting the electrons through the very thin samples. Okay, instead of light, it will uses the electrons. It will allows the researchers and scientists to examine the structure and composition of various material at atomic level. Okay, main advantages, it is having the ability to provide detailed information about material being studied. It is possible to determine crystalline structure, chemical composition and even the arrangement of individual atoms within it. Okay, this term is widely used in the field of material science, biology. For example, it has been instrumental in developing the new materials for applications ranging from high strength alloys to advanced electronics. Okay, it also used to study the biological structure of cells in more detail. Okay, the use of this term requires highly specialized skill and precise conditions to achieve the optimal result. Especially the sample should be very thin, which will be in the size range of few nanometers only. Okay, the next one is scanning electron microscope. It is used to capture the high resolution images of a surface of a sample. Okay, it will works by detecting and amplifying the signals generated by the electrons that are emitted from the sample during imaging. It is mainly used in the field of science, material science, biology and nanotechnology. Okay, it also uses a focused beam of electron towards a sample, but it will detect the electrons that bounce off from the surface of the sample, and it will be captured by the detector. Then the signal generated by the electrons is then amplified and used to create an image of the sample surface. Okay, the sample is mounted on a stage that is placed within the sun chamber. The stage moves the sample, which will allow the operator to scan different parts of the sample surface. It also equipped with a magnetic lens, which helps to focus the electron beam. It is widely used in material science for identifying the structure of material, then in biology for visualizing the cells and tissues, in nanotechnology for viewing the nanoparticles. Then main advantage of SEM is high resolution imaging, then non-destructive analysis, which means it will does not damage the samples, then versatile, which means it is used to analyze wide range of samples. Then disadvantage include it is very expensive, then the sample needed to prepare carefully for some analysis, and this process may be time consuming. Thank you for listening.